Hello, I'm Ilham Lomari, and this is my graduate seminar. I'm gonna talk to you about influence of temperature stress on immunity of honeybees, Apis mellifera. Honeybee, Apis mellifera, is a highly eusocial insect which considered one of the most important pollinators worldwide. Without bees, a 90% reduction in crop yield is expected. Today we are gonna talk about one of the most important threats that are facing honeybees, heat stress. Environmental stresses, especially temperature, affect most organisms, including honeybees. Stressed individuals are less productive and deliver fewer resources to the hive, so they will have lower priority in getting any of the group resources, which is already limited. Immune activation, which is associated with stress, will alter learning behavior, individual and social behavior also. This will result in less contribution from individuals to colony. On the other hand, temperature shock, as a stress, have a good effect on immunity for the next generation of insects. Insects' immune system helps them to be more than the half of living things on this planet. Defense mechanisms of insects divided into humoral immunity, where humoral means body fluids. The main criteria for this part of immunity is the synthesis and secretion of antimicrobial peptides, AMPs. Cellular immunity. The responsible for this part of immunity is the free blood cells. Insects have within their open body cavity free blood cells called homocytes. The heat shock reaction pathway, HSR. Heat shock or temperature stress as we know it will induce a reaction in cells that is called heat shock reaction. In this reaction, cells will secrete heat shock proteins, which is normally produced in low amounts in cells. Heat shock protein maintain balance of protein synthesis, folding, functioning, and degradation in many organisms. Heat shock proteins also causes immune effectors and antimicrobial peptides gene depression. Heat shock response might have unique properties in honeybees due to their particular lifestyle and exposure to significant routine stresses. Heat shock response and humoral immunity response are antagonistic in honeybees. Such a relation is explained by trade-off theory. As we know, individual bees have limited energy resources, so trade-off between either HSR or humoral immunity is necessary to achieve the best result for the colony. The effect of external factor on the host-pathogen interaction is illustrated in this model. In this model, we will refer to honeybees as the host. During their lifetime, honeybees faces many stresses such as external environmental conditions, which include temperature. Such a stress will influence the immune response for the host by activating signaling pathway, which will influence the defense mechanisms against pathogen. Those same external conditions will affect the virulence of a pathogen, either by increasing it or lowering. So, as a total, environmental conditions could have both positive or negative effect on either host or pathogen. Acetylcholinesterase is an enzyme that catalyzes neurotransmitters as acetylcholine. This process will terminate synaptic transmission. Honeybees' acetylcholinesterase is induced by heat shock along with many different stresses also. Organophosphorus and carbamate insecticide toxicity increases with temperature increase, but honeybees' acetylcholine esterase increases will provide protection against toxic chemicals, particularly under heat stress. The negative side for this alleviation is that acetylcholine concentration will be reduced, resulting in downregulation of immune response for honeybees. As shown in this table, heat shock as a type of stress will induce expression of acetylcholinesterase in both head and abdomen of a bee. Another marker gene, which is considered important, velitogenine, VG. This marker gene will increase by heat shock also. Velitogenine is considered as an antioxidant. Its increase will enhance immunity, but as we can see in the same table, it will reduce in cold stress, resulting in less immunity levels. We should keep in mind that all of this is controlled by trade-offs theory for the benefit of the colony. Varroa is one of the most important ectoparasitic pests that attack honeybees and feed by sucking hemolymph. Such a process will elicit transcriptional response which give higher heat shock protein. Varroa interfere with thermoregulation process which could affect winter cluster leading to a higher thermal fluctuation. Nosema, a common disease of honeybees caused by Nosema carnica and Nosema apis. Infection with Nosema increases with temperature increase. Remember that heat shock response and humoral immunity are antagonistic in honeybees. Jackbrood disease is a fungal disease that infects larval stages of bees. Recent researches showed that both high and low temperature increase the expression of immune response genes in leaf-cutting bee and reduce the level of the disease. Honeybees during winter remain in clusters inside the hive. They produce heat by a process called shivering. This process is energy consuming. We should always remember that bees depend mainly on stored food as a honey in the colony. If we compare winter bees with summer bees, we will notice a reduction in expression of genes involved in the cellular immune response and physiological activities. We will also notice that winter bees maintain a high expression of humoral immune genes involved in antibacterial defenses. Temperature decline could lead to an increase in severity in the viral infection in emerged bees. Most losses of the colony occur during winter. This observation suggests that winter bees may have compromised immune functions and higher disease susceptibility. How can we explain that? First explanation is adaptation to environment, which bring back trade-off theory. Low resources force reduction in costly immune and physiological activities. The second explanation is the evolutionary adaptation. The expression of some immune genes in winter reflect past evolutionary adaptations to pathogens. Finally, we should keep in mind trade-off theory, where balance is created between resources for the benefit of the whole colony, even if that leads to reduction in immunity. Also, sublethal heat or cold will reduce immunity, leading to higher viral infection rates. Some diseases, as Nosema, will reduce immunity also, and they are widely spread during high temperature. All of that will lead to higher mortality rate and colony collapse will occur. Thank you very much. Any questions, you are more than welcome. Please leave it in comments and I will answer it as soon as I could.